So good afternoon. My name is Sherry Xie. I'm currently a bioinformatician in Dr. Avi Mayan's lab here at the Icahn School of Medicine at Mount Sinai. And today I'm just going to be talking to you guys about clustering algorithms. So as kind of an overview of today's lecture, we'll go over a few commonly seen algorithms, especially within the biomedical research space. And this includes k-means, dbScan, the Leiden clustering, and then hierarchical clustering and weighted gene co-expression network analysis. But before we get into the actual clustering algorithms, kind of as an introduction for why we even want to do cluster analysis in the first place, we know that biological data is often very high dimensional and also very interrelated. So if we think about gene expression data from cancer samples, you have maybe 20,000 plus unique gene measurements per sample, so it's very high dimensional. And then all the samples are related to each other because it's likely that they overlap or are variable across very different metadata categories. And you kind of want to look at the patterns across those different samples. And then, of course, we know that the expression of individual genes is not independent. So that makes it even more important that we're able to kind of look at these patterns across each of these different gene expression profiles. So that leaves us with this question of needing to quantify the relatedness of the data that we have. In some sense, we're looking at what genetic or clinical features might be driving the similarities or differences between samples. And then for more translational perspective, maybe how do they impact things like patient prognosis, or could they provide insights into future therapeutic interventions? And so it might seem kind of intuitive to kind of by eye look at the groupings in this data. But your intuition will only get you to a certain point. For instance, we saw earlier that those two large high density clusters on the left and the right side of this plot, which is just a PCA plot of papillary adenocarcinoma samples from the Cancer Genome Atlas. Those look like they might be clusters, so like very related samples. But then once we get to this chunk of data samples in the middle, we have a harder time actually telling if this is part of maybe the cluster on the right, if it's its own independent cluster, or if it's multiple smaller clusters. And so that's why we need cluster analysis to be able to have a method to kind of tell us where these clusters might actually reside in our data. And it's important to note that clustering is an unsupervised machine learning method. And it's unsupervised because generally we don't actually know the true clusters. So cluster analysis kind of just provides a way of systematically determining what are likely clusters. And most algorithms will actually look at both similarities in the features within a cluster, as well as the differences between clusters. So basically making sure that clusters within a cluster, you get samples that are very similar to each other. And then those samples are very different from other clusters. And pre-processing and normalizing your data will also affect clustering. So that's something also to keep in mind, but you'll hear more about that in next week's lecture. There are two main types of clustering algorithms. So there's partitioning methods, and these basically identify the best divisions of clusters. So you can think of like where to draw the lines on a plot. And in this case, clusters are non-overlapping, and you can think of this as maybe like distinct cancer subtypes within your data, or if there's some demographic category, like sex or ethnicity, where you can have these very discrete categories. Partitioning methods would model the data pretty well. On the other hand, hierarchical clustering builds a tree of nested clusters, and I'll discuss this more in depth once we get to the section in this lecture. But of course, in hierarchical clustering, you're necessarily building overlapping clusters, and this might be more appropriate for modeling things where you know that what you're looking for might be like subgroups within larger groups. So perhaps like sub-processes of certain pathways.